Hello everyone. This is Shivaranjani Koti from RVSKVV, India. So first of all, I would like to thank the organizers for organizing this wonderful symposium and giving us a great opportunity to present. So let's move on to the title of my presentation, that is impact of temperature on variable isolates of R. Bataticola causing dry root rot in chickpea. So let's move on to the background. Crop production. Crop production generally aims to maximize the yield to meet the global population demand of the global population. But due to certain reasons, there might be a decrease in crop production. The main reason behind it is the pest and disease outbreak that occurs due to the climate change, which is the burning topic nowadays, due to which there is increasing temperature and CO2 levels in the atmosphere. So now in my work, I'm going to study whether this temperature is affecting our Bataticola or not. So let me give a quick introduction about the disease and the pathogen. This is the infected field. When we see from the distance, we can see a patchy or scattered appearance of infected and healthy plants, and this is how the individual plant look like. And when we pull out from the ground, we can see primary root, which is divide of secondary and tertiary roots. When we split open this root, we can see lots of sclerotia present inside the pith region. When this pathogen is grown in the culture, this is how the culture looks like. This is the fluffy texture. Along with this, we can see a cottony and oppressed growth as well. When this hyphae is seen under microscope, you can see the right angle branching, which is the characteristic feature of this pathogen, and the sclerotia under 10x is how it looks. We move on to the objectives. The first objective is all about the in vitro studies, how the temperature affects culture and morphologically on Rhizoctonia bataticola. And second objective is how the temperature affects the disease expression. In third objective, we'll study how fungal biomass colonization is affected by different contrasting temperatures. So these are the methods I followed all throughout my experiments. In first objective, I have taken 30 isolates, out of which 15 were collected from different districts of Madhya Pradesh, and the rest 15 were taken from different states of India. And these are the temperature regimes I have used all throughout my experiment with 3 replications each. And the media we have used is potato dextrose agar media. And these are the parameters I have recorded. A radial growth I have recorded after 48 hours of inoculation, and the rest parameters like colony texture, aerial mycelium, and spheroidal time, intensity, size, shape were recorded after 7 days of inoculation. So the results obtained I will be expanding in the coming slides. Now we move on to the method followed in objective 2. In objective 2 also I have used 30 isolates with the same temperature regimes with 3 replications each. But here we followed paper towel technique. In this technique, 7-day-old chickpea seedlings were taken and were dipped in a macerated inoculum for 2 minutes, and then we have placed those seedlings inside the paper in the tray, and we shifted those tray into the incubators at a different temperatures, that is starting from 20 degrees to 40 degrees centigrade. And after 7 days, from 3rd, 5th, and 7th day after inoculation, disease severity rating scale was recorded. Based upon this modified disease rating scale, and move on to the objective 3. Unlike 1 and 2 objectives, here I didn't work with all the isolates. I have taken a few isolates, the highly pathogenic ones, and I have mass multiplied those isolates in the sand maize media, through which I have made 6 soil out of it. After making 6 soil, I have sown BG212 seeds and placed those pots in 2 different contrasting temperatures, that is 20 degrees and 30 degrees centigrade. After 15, 25 days, 35 days after sowing, I have taken data on disease susceptibility index, and I have collected root samples after each observation, from which I have extracted DNA, and we have used the DNA for few PCR studies. so that we can find out how much nanogram of pathogen DNA is present inside the root tissue. So these are the results obtained in the objective one. The radial growth I have mentioned, graph is shown here, which is taken after 48 hours of inoculation. And in the x-axis, I have placed the isolates. In the y-axis indicates the diameter in mm. Different colors indicates different temperature regimes. We can see as the temperature increasing, there is increase in the diameter. This horizontal line denotes they're drawn by taking the arithmetic mean of all the isolates along with the replications. So keeping these aside, other parameters like colony texture didn't show any difference with respect to temperature, but few isolates I have observed showed the colony texture change from cottony to fluffy and from fluffy to the pressed growth. And while moving to the second objective, that is disease severity, you can see how on this basis I have given the rating. You can see the control plant with the rating scale 0. You can see the absolutely healthy roots with lots of secondary and tertiary roots. And move on to the scale 1, you can see some sort of lesions, but you can see good amount of secondary roots. When we move on to scale 2, there is thinning of roots with some more lesions, but here you can see very little amount of secondary roots. And 3 and 4 you can see, in fourth one you can see almost depth of the seedling. So I have rated according to this as such. So when we see the this heat map, here I have to make you see understand easily. I have placed these in the heat map. When we see with respect to temperature, as the temperature is moving from 20 to 35 degrees, you can see more disease severity, that is 4 degrees centigrade you can observe. Whereas as the day is progressing from 3 to 5 and 5 to 7 also, you can see significant increase in rating scale. And move on to the objective 3. DSI as well as pathogen colonization was shown here. With respect to DSI, you can see there is increasing disease susceptibility as the time points increasing, and with respect to temperature, significantly, like you can see more disease susceptibility at 30 degrees centigrade as compared to 20 degrees centigrade. This pattern followed in all the 3 isolates. We can see RB6, 24, and 28 as well. But when we move on to the colonization, pathogen colonization, you can see the same as the time points increasing, there is increase in colonization. But with respect to temperature, in first 2 isolates, more colonization was observed at 20 degrees. But in RB28, you can see more colonization at 30 degrees centigrade. So from this, we can say the temperature do not show any impact on the colonization, but you can temperature impacts on the expression. This is the expression. That means more disease is observed at 30 degrees, although the colonization is similar. So conclusion, from this we can conclude that in both in vitro and in vivo conditions, the optimum temperature for pathogen growth and severity is 30 and 35 degrees centigrade, and both above and below reduce the growth and severity as well. At higher degrees centigrade, the pathogen failed to grow, and even the disease severity also, the plant unable to survive due to this excess heat. Like 40 degrees is too much for continuing the physiological condition, like what conditions. And colonization, with respect to colonization, no much difference is observed with respect to temperature, but this is susceptibility is more at 30 degrees centigrade. So from this, we can say that we can adjust the sowing dates in such a way that peak flowering stage should not be coincided with high temperatures. Thank you. Climate change is no longer so far a problem. Because of this, we can say this pathogen is now emerging, causing great damages to the field. Thank you.